Ladies and gentlemen, welcome to the afternoon session and to a very special event that we are very urgent to get out before 3 o'clock because the speaker wants to, I don't know what he wanted to do. There's something at 3 that he wanted to do. Anyway, the ISCB Senior Scientist Award, the awards committee chair is chaired by Bonnie Berger. This statute says the Senior Scientist Accomplishment Award recognizes members of the computational biology community who have made major contributions to the field of computational biology through research, education, service, or the combination of the three. Here are our previous senior scientist winners. All of you will recognize names, all of you will recognize textbooks, all of you will recognize when you learn computational biology. Those are really founding fathers of the family. These founding fathers, amongst them, have published over 3,500 papers with an H index of over 170. That's ISI by July 2014. And they acquired all that through their excellence and outstanding scientific achievements. And this is the moment when Wagner should play the Valkyrie, because that is obviously the way I learned Gene has motivated his gang to assemble the human genome, but somehow iTunes doesn't have the permission to play the Valkyrie. Gene Myers, currently at the Max Planck Institute in Dresden, Germany, born in Idaho, did his PhD at Colorado, became a professor at the University of Arizona, Tucson, became vice president at Celera, professor at Berkeley, went to HHMI, Howard Hughes Medical Institute, Genelia Farm, and then went to the Max Planck Institute in Dresden. One way of, and somehow my machine hangs. This is too much to put into a single slide. That's why my machine hangs. I'm not sure what it is. So one way of looking at Gene is what are his most important contributions. In 1990, a suffix tree data structure. So think about suffix trees and then think about Gene. That's one thing he does, or did in 1990. And that was at the heart of BLAST. In 2000, he assembled the human genome. 10 years later, something that is not quite entirely related to the same thing, but influences us as much as the first one. 2010, image of the fly brain, another discipline. So we clearly have a man here who every 10 years needs to change the world with a different subject because the grandeur of what he's doing, there's nothing else he can do in the discipline. The image of the fly brain, and he will talk about how that influences us today. H index, 52, 9 papers quoted over 1,000 times, 35 over 100, over 180 publications. And I'm sorry, I have an issue with my machine. Now at the Max Planck Institute, he's going to talk about that. In the Max Planck Institute, he has the list of his top 5 papers. And 4 of those are, you can bet in which journals, or you can guess in which journals they are published. One of them has been published at the ISMB, ISCB in Vienna. And that is another aspect of Gene. Gene is, as he says, a doer, a technologist, an engineer, somebody who changes the world by doing certain things, by creating things. And he roots in this community. And this is who we are. We produce the right methods, not just analyze data, but we produce methods that make a difference. And Gene, more than anybody else here today maybe, has made that happen through his work. I'm very, very honored to introduce Gene Myers. And there's this one thing before I let him talk. So Gene, the point is I couldn't do this iTunes thing. Can you just sing it?
No. Dun, 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 dun. It's the, at Caltech, at the first day of finals week, they used to play the Ride of the Valkyries at full blast. And it was meant to instill fear into your being. And that's kind of the way Celera felt for the first, actually, that's the way Celera felt for the entire time. And so what my group used to do is we used to play the Ride of the Valkyries on Monday morning and have a big Nerf battle in order to try to get the willies out and get to work. So I'm actually going to talk about assembly today, but I figured this is probably one of my few award talks. So if you'll forgive me, I'm just going to do like 3 vanity slides where I talk a little bit about my overall career. Because I divide my career into 3 epochs. And the first epoch was kind of the early days where basically I was a traditional computer scientist. And I thought about algorithms. There I am standing in front of my spiffy macmolecule software in my, I thought about mathematical things, lemmas, theorems. And these are maybe some of the things already, suffix arrays have been pointed out, but Webb and I popularized the idea of computing alignments or delivering alignments in linear space. And also I did some of the, I was involved in the BLAST search engine. And kind of one of the reasons I wanted to do this slide is that recently, last year I wrote a paper about what's behind BLAST. In other words, the theoretical underpinnings, because there's a piece of work that I did that I don't think a lot of people know. While BLAST has been cited 50,000 times, the actual theoretical piece that led to BLAST has been cited 240 times, 40 times by myself, of course. So I thought you might want to go look at that paper. If you Google what's behind BLAST, you'll get to the PDF, okay? It's a historical piece and a technical piece. And then if you don't want to read the paper, you can Google Yandex, which Yandex is the Russian Google. You can Google Yandex, what's behind BLAST, and it'll actually take you to a video of me giving a talk about that paper. Okay, so I thought you guys might enjoy that, just in case I blow this talk, okay? That one's a good one. This is my, so I'll skip to the end of my career, so there I am with significantly more gray hair in front of the Blaues Wunder. And I just wanted to convey that you know, so the interesting thing is I went from a theoretician to this guy now who's building these microscopes that you can see in a couple of the pictures. And I'm really fascinated by development and basically the mesoscale of the cell as a physical system. And I really think that that's a very kind of interesting frontier. And you know, I'm hoping to make some contributions in the area of technology there. But that brings me then to the middle period, okay, which is really kind of where I made the transition. It was really that kind of time at Celera where I was involved in the company and in the technology that really allowed me to transfer from being really a theoretician to being somebody who's more pragmatically oriented when I realized that, hey, I can really think about technology and I can direct technological work. And it was really there working at that level that I was able to stretch myself. And so there we are. Those are the Valkyrians that I worked with at Celera 1. And these were the kinds of things that I thought about, okay? String graphs, data flow, and these are some of the things that we did. And today I'm going to talk about that because it's basically, it's been 30 years since I first started working in assembly, and I thought it'd make it good. And it's been a problem that I love and that I've been thinking about for my entire career. And in fact, I've actually started working on it again, okay, in the last year. So I've been at it for 30 years now. So I'm going to cover the period from 1980 to now, okay, at some level. And so this picture that I showed you was the Celera floor in 1999. And kind of the punchline is going to be what I call the resurrection of de novo assembly. And I'll try to say some things that are mildly provocative as I go along to keep it interesting for you guys. Okay, so who did the first shotgun project? Fred Sanger. It's no wonder this guy won 2 Nobel Prizes. So the first shotgun was the lambda phage. It was the very first kind of whole organism that was sequenced. It was the whole organism. And it's, you know, I looked at that paper and I was just boggled by the number of ideas in that paper. So for the sampling, they went from restriction enzymes, realized restriction enzymes weren't random enough, so they went to DNase 1, realized they had problems with the temperature, and went all the way to sonication, which then prevailed for a long time until people got to nebulation. They also wrote about gap finishing, DITP for GC-rich regions. He gave 2 ways of doing mate pair sequencing, okay? He gave the idea of sequencing progressive deletions and of probing a library to find the gap-covering clones. This is all in one little paper, you know, in the 1980s style. And then finally, this was the beginning of software for assembly, and it was Roger Stodden who basically wrote a program that would find the overlaps. And it didn't even use dynamic programming, okay? It was basically a greedy-based way, but it would basically find them, and then it would put together the reads, but it didn't really commit to it. You basically had to solve the puzzle yourself. So it was very manually oriented. So what happened then in about the 85 to 90 period, I would call the computer scientists coming in. And so actually, Esko Ukkonen was the first person to come in, and he developed a little assembler called Seek8. It was very modest. I don't think it actually was used by anybody, but it was a beautiful little paper. And you know, it's actually, a lot of people credit me with OLC, but really all I can take credit for is calling them overlap, layout, and consensus. You can already see the idea of these phases, right, in Esko's paper. And in fact, there's this kind of schism in the community between, I'm building an OLC assembler and I'm building a de Bruin assembler. And I would actually argue that a de Bruin assembler is an OLC assembler too. Building the graph is overlap, okay? Disambiguating the paths using the reads is layout, okay? And then you do consensus at the end. I mean, it's basically, there's really only one way to skin this cat, okay? Except there are different ways to actually do the individual things. The next thing that happened is that, along with Jonathan Turner shown there, is that the theoreticians kind of said, it's the shortest common superstring problem. I want to find the string that's as short as possible that explains all the data. And this was a nice computer science problem, NP hard, and these guys worked hard on approximations that would solve the problem. Then John Kececioglu, my student, first student actually, and I basically were also kind of working on this problem, and we basically kind of got more high-powered about some of the OLC stages and how to solve each one of these problems in a more pragmatic way and in a more sophisticated way. And I think probably one of the nicest things that that came up was this idea of a bidirected graph to model the orientation, because I still see people get confused about this. You know, the fact that really DNA is a duplex, and so you get reads from the 5' strand and reads from the 3' strand, right? And so there's this issue that you have to think about orientation, and there's really a nice, just kind of to throw a little mathematics into the talk, right? So it's not just all opinion, is think about just ignoring all the reads that are contained in others, so that you're left with just a set of reads that properly overlap each other. And if you think about it, there are basically 3 cases, okay? So there's the case where the 2 reads are in the same orientation, they're in the opposite orientation, and they're kind of facing each other. The 3' ends intersect each other, and here the 5' ends intersect each other. So what I'm going to do is I'm going to put a yellow ball on the 5' end and a green ball on the 3' end, okay? See where the balls are? Okay, balls are in different positions. Now take each read and turn it into a vertex, okay? And notice that I've put the yellow and green balls in them, and I've been careful to keep the orientation, right? Yellow, green, yellow, green, yellow, green, yellow, green. This guy, green, yellow, green, yellow. And then what you do is you put an edge like you would in a normal graph, except this graph is a funky graph. It's got an arrowhead at both ends of the edge. And the arrowheads can go in different directions. They can go towards the node or they can go away from the node, okay? So bi-directed, okay? And the rule is always point in away from the green one and point in to the yellow one. Okay, if that's the side you're on, okay? So away from green into yellow, away from green, into yellow, into yellow. I call this a normie, this is an innie, and this is an outie, okay? So what does that buy you? Well, let's think about a real simple example. A, B, C, D, where C is reversed, okay, with respect to the other 2. If you build that if you build a set of sequence of edges, there it is, okay? Now, take a look at like vertex B. You have 1 arrowhead in and you have 1 arrowhead out. Take a look at vertex C. You have 1 arrowhead out and 1 arrowhead in. In fact, what I claim is any path where the definition of a path is in degree 1 and out degree 1, right? Just the standard normal graph theoretic definition of a path on a regular directed graph, that represents an assembly, an appropriate layout or path, right? And so that solves the problem. Okay, so let's never discuss this again. Okay, all right? Okay, because I actually in, I think, 2003, when Ewan Burney was doing Velvet, I think Velvet is his thing, he was thinking, I'm really confused about how to do the complement thing, right? I mean, because it's usually not talked about, right? So I thought I'd put that one to rest. Okay, so I think the kind of the coolest part of the story, actually, is that at exactly the same time, in exactly the same place, exactly 1 hour apart, there were 2 talks given, okay, which basically both introduced the major string graph paradigms, the Bruin graph and my unitig graph. And in fact, I think what a lot of people like, so Pavel is the guy who's really done the most work with the de Bruin graph, right? But it was actually Waterman and Aduri at the DIMAX meeting in 1994. It was Aduri who got up and introduced the idea of a de Bruin graph assembler, okay? The origin was that Waterman was looking at Pavel, and Pavel was thinking a lot about sequencing by hybridization, right? Remember, the plan was to use what were the AFI chips, right? The AFI chips are actually a failed sequencing chip. I think that's one thing that people don't realize is Dramatic was trying to do DNA sequencing, and they couldn't figure it out. And Pat Brown came along and figured, I can use this to do expression analysis. Horrible sequencing chip, not accurate enough for Kamer binding, but good enough for expression analysis, okay? So, but Pavel had done a lot of the early work, and he was using de Bruin graphs, but it was Mike who kind of went, assembly, okay? And so here you go, here's a de Bruin graph. I don't think I need to explain. This is a Bruin graph of 3 mers. You basically take every k-1 mer, and then you connect them together. I'll actually do the construction for you in a minute. But here's the cool thing is that Mike says, well, after I kind of disambiguate all the reads, I might get something like this. So if I have a piece of DNA that has 3 copies of this repeat R, then I'm going to get this graph. Okay, right? It's a string graph, right? You guys are all used to this now, okay? But this is the first drawing, okay, of such a thing, 1994, okay? Now, Erduri gave his talk at, I think, 1 o'clock, okay? But at 11:30 that morning, this other guy, Myers, got up, and at the same meeting got up and talked about a unitig graph assembly, okay? And it was really about chordal graph assembling. And there were 2 points about the talk. One was that this whole thing about shortest common superstring was wrong. That when you did shortest common superstring, you were over-compressing the reads. And so I actually gave a formulation where, look, the real problem is to find the maximum likelihood solution with respect to the statistics of coverage on the assumption that you know that read arrivals are Poisson, okay? But they have to be Poisson. And so here's a picture showing how it's messed up if you do SCS. But then I give this chordal graph collapsing idea, right? And I wasn't fully committed at the time, but you can see the stages, and look at this graph down here. Gee, it looks a lot like the other one, right? And in fact, it is exactly the same thing, except I only did 2 repeats. Mike upstaged me with 3. Okay, I just did 2, but anyway, but you get the idea, right? So I thought that was really a kind of magical moment. I remember watching Aduri's talk and going, Jesus, okay, it's like there's this consilience, right? So here are these talks, and so in fact, Mike was calling his a sequence graph, and I think I was calling mine actually a chunk graph. Okay, you know me, I come up with really hokey names. And what I want to explain to you is that they're both what I call epsilon string graphs. Okay, they're really like 2 sides of the same coin. And so the idea of a string graph with a set of reads is it's basically think of a graph. So here's a graph, and imagine that every one of those threads is a DNA strand that has letters spelled along it. Okay, and the idea is you want 2 properties. You want that every read in your data set approximately matches, okay, some arc of that curve, right? Some path through that curve. It epsilon matches, okay? So the epsilon is meant to take care of errors in the data, that it epsilon matches to a segment of the read. And then you want it to be complete, and I'm sorry, I had PowerPoint fatigue, but I should have probably drawn every read covering it. But basically, every part of the graph should have at least 1 read binding to it, right, or matching it at epsilon, okay? And the interesting thing about string graphs is, in some sense, and I didn't really kind of realize this later, but if you think about it, everybody's kind of like, well, the answer to assembly is like this linear string. And what I realized is that no, if you have a set of reads and you've got a certain amount of information, then what theoretically is the best answer I can give you? So imagine that this is a genome, okay? And imagine that the yellow parts are completely unique, okay? And the two green things, the three green things are repeats of each other that you're going to mix up at your error rate. The purple things are two copies, and there's two copies of an orange repeat, okay? Then what I claim is that in some sense, the best possible answer is this thing, right? If the repeats are longer than the reads. So imagine the reads are short. So those repeats, those colored arrows are big. Okay, they're 10,000 base pairs and your reads are 500 base pairs. Then, in some sense, you're not going to resolve the order of those repeats, right? But you can build that graph, okay? And in fact, you can do more with that graph. If you know that the reads have been well sampled, relatively uniformly sampled, you don't have a strong sampling bias, then you can just use simple statistics to estimate that the yellow parts are actually single copy. And then once you know that those are single copies, then doing just simply a simple graph analysis, knowing that the answer needs to be some Eulerian tour of this graph, right? You know the copy numbers of the repeat parts, right? Because there's 2 ways in and 2 ways out here, 3 ways in, 3 ways out there. And in fact, you know then that some Eulerian tour that observes the copy numbers must be the answer. And it turns out there's only 2. Okay, those are them. So see how far you get? I mean, okay, if you could build this graph, right? And so I think everybody's onto this now, okay? Everybody, but at the time this was kind of cool stuff. So in some sense, a string graph resolved at the levels of reads, in other words, you've used all the information in the reads, is in some sense the best possible answer, absent any other information like mate pairs or an STS map or any of that kind of stuff. So, I mean, basically, I think I'm going to just rush through this because I don't really, but look, because either you know how to do this or you probably don't care. But the point of the matter is that a Bruin graph, okay, is a zero string graph. In other words, it's a string graph in which there are no errors. All the matching is exact. And it's a string graph over the k-mers. It's what I call deterministic. The big pro about the Bruin graphs is you can build them in linear time. Okay, but there are some cons, okay, which is that any kind of variation or error shows as billions of bulges in the graph. Okay, so it's not really talked about very much, but for example, if you're building a de Bruin graph with respect to 20-mers, if you have a 1 base pair error in a read, that will create a bulge of 20 characters, not 1 character, not just 1 little character, 20, okay, the size of your k-mer. And so you get these bulges all over the place, okay? And so what people do, and it was kind of buried, frankly, in the first paper, is you take all the k-mers that don't occur too often, and you suppress them. You say, oh, those are erroneous k-mers. So in fact, what you're doing with the de Bruin graph is you're doing a form of error correction. You're saying these k-mers are wrong. Okay, get rid of them. And if you get rid of them, then you get rid of most of the bulges, the false bulges. Okay? And it's a little secret, right? Everybody removes the k-mer that occurs 2 or 3 times, right, in a 10x data set. The other thing to note about these, which will be important for the end of the story, is that if you have very, very high error rates, it doesn't work, because if you have low error rates, then the consensus sequence, which I show in red there, actually will end up showing up. But for example, in the case of PacBio, the number of conserved k-mers is so low that you don't see the consensus. There aren't enough conserved k-mers to actually see the consensus. You just get nothing but bulges, okay? The other con is that you then have to basically take this graph and you have to figure out what the reads say, right? So you have to you then go into the stage where you say, oh, that path can't possibly be because there's no read that supports it, okay? And so you spend a lot of time trying to resolve the reads, okay? So, and don't worry, there are cons to the string graph as well, okay? So the unitig graph is an epsilon string graph, and so, I connect the nodes as you indicated. Here's the interesting thing. You have to do a little bit of work to get a unitig graph, which is suppose that this is a set of reads here, right? And right at this point, you can see there's a divergence, right? So that the two sets of reads start spelling something different. If you build a string graph, if you find all the overlaps, you get all these edges, including these purple ones, which are in some sense transitive, right? Because this guy overlaps this guy and then overlaps this guy, and that pair of overlaps implies this overlap, right? So the thing that you do is you do basically transitive edge removal, classic transitive edge removal in the graph, and you end up getting what I call a unity graph, okay? I call it chordal graph collapsing, okay? And the point of the matter is that that is an epsilon string graph of the data set as well. I won't prove it, but it is, okay? And the pro is that it's mostly resolved at the levels of reads. In other words, you don't actually have to do this thing where you try to figure out what the read says about the paths through the graph, because you've built the graph at the level of resolution of reads, okay? So once you've got the graph, it's a much sparser data structure, right? Because the number of nodes is proportional to the number of reads, not the number of k-mers, okay? But the big con is that if there are repeats in the sequence, the thing takes a quadratic amount of time, okay? And nobody likes that, okay? All right. Okay, so that was, so I thought that was, so the next thing then is I guess it gets a little bit personal, but it is what I call the shotgun era, okay? And I drew, you know, the 3700 capillary gel sequencer in all its glory, okay? Just in case you've forgotten it. And it was in 1998, it was pretty cool. But I want to, the story for me really starts in 95, and I want to talk about this guy, Fred Blattner, who's actually responsible for your E. coli K12 sequence. Okay, and Fred was kind of a maverick, a cool guy, but a little bit off the beaten path. But I went to Wisconsin and hung out with him one time, and he said, Gene, I really want to understand, like, you know, how many contigs will I have where there are 2 reads at least covering every base in this direction and at least 1 base in the other direction. Okay, so what I had to do is I had to take the Lander-Waterman-Poisson analysis and really go to town with it, right, and understand it and figure out how to generalize it. And so I spent, thanks to Fred, I actually ended up spending, you know, quite a bit of time thinking about that analysis. And, you know, so, and most people used that analysis at the time to say as a rationale for why whole genome shotgun wouldn't possibly work. And you had to do backs because, you know, hey, if you had 500 base pair reads, which was a long read at that time, and you did a 6-fold coverage, then if you sequenced a back to 6x, then you'd get like 4 to 5 contigs, right? And that was okay. But if you did it on the human, you'd get 104,000 gaps. Like, that's horrible. And I don't know the order and orientation of those, right? So we can't do that, right? It's not good. But basically the number of gaps grows linearly with the genome. And the truth of the matter is that if you sequence 25,000 backs, okay, which is what it takes to get a human genome, and they each have 4 errors in them, then you're going to have 100,000 gaps in all of your backs. So I wasn't, you know, and that thought kind of hit me, and I went, you know, hey, okay, what exactly are you guys talking about, okay? And in fact, if you look at other things, like how much of the object is going to be covered, exactly the same. How big are the contigs going to be? Exactly the same, okay? How big are the gaps going to be? Exactly the same, okay? Everything's the same. In fact, to kind of put it in other words, if I take a back and I sequence it at 10x, and I put down all the reads, that's what I get, okay? Now imagine that I sequence the entire genome at 10x, and I take out that back, okay, and suppose I have the same random number generator, and I looked at how it would be covered, okay? The same thing, okay? So, but of course, this is a bit simplistic because it says that I know where the reads go, right? And of course, the real reason that people were worried about this is that there are enough repeats, right? That you don't really know where to put things, okay? And that's really the problem. And also, that people wanted order and orientation. So having a bag of 104,000 contigs wasn't enough, okay? But then Jim Weber and I had a phone conversation in '96 where Jim said, well, how about we do mate pairs of everything, okay? And so I really started to think about what the power of mate pairs was, okay? And I think this is really where it comes in. And in fact, we wrote a very controversial perspective in '97 that it's the only piece that I've ever written that was immediately followed by a counter rebuttal of equal length by Phil Green, in which Phil gave a brilliant argument for why his assembler would not achieve the job, in my not so humble opinion. we did a tremendous number of simulations, okay? And you can see I already had the idea of a scaffold, okay? And it actually, a full paper, I was not allowed to publish the full paper. A full paper actually appeared here, in case you're interested. And you can find that on my website. But look, what we showed was the following, which I don't think anybody really understood in that paper, which is that if you could detect which reads belong to unique DNA and which ones did not, that you could, in principle, resolve 99+% of the genome, okay, using the mate pairs, okay? And it was definitely a theoretical piece. I mean, I hadn't done it. I'd just shown that from an information theoretic point of view, if the genome consisted of what the known composition of line elements and sign elements, of the known interspersed elements, that they would not pose a problem as long as I had 10,000 base pair mate pairs. And so the idea was to go forward with this. Now, the interesting thing is that we proposed this and nobody wanted to do it, okay? I was cruising Europe trying to see if I could get, forgive my German colleagues, I was trying to see if I could get the Germans to do something modest, okay, with also to no avail. And, but then in 1998, Celera was formed. And it was like, and you know, they were going to do it my way. So I joined them, okay? I had no choice, if you really think about it, okay? Intellectually, I had no choice. It was the only place I was ever going to get a chance. So I went. And of course, this was said, and everybody thought it was going to be the Swiss cheese. I don't think I want to actually do this part about the other thing, but basically the human plan, the HGB program was basically taking the genome, turning it into a physical map of backs. In other words, they're doing a pre-assembly of backs based on fingerprinting. They then pick out a tiling set, a nice tiling set, and they shotgun sequence those individual backs because they felt like, no, I got to sequence backs. So, but if you think about this from a factory point of view, it had the drawback that you had two separate processes. Okay, you had to have the STS map or the back map factory, and you had to have the shotgun factory. So you had two separate factories because the technologies did not, the two processes did not share any common technology. The other problem is that the clones weren't stable. They were mutating all the time. Backs are not stable. So you actually, even if you built the map, you couldn't be sure you had what you had when you put it in the freezer a month ago. But the good points was that the cost was paid, and they were spending your tax dollars, and they could guarantee, okay, that they were going to give you a result. Which is, you know, if I was in the same position, I might have done the same thing, right? I mean, you know, if I was going to be held to account. But fortunately, I was in a place where I wasn't held to account, okay? We had a lot of money, okay? Craig was saying kind of crazy things and telling everybody what we were doing before I had done it, okay? But I was having fun in the background, and the plan was to basically do these paired ends, okay? And to do this big shotgun and to produce these scaffolds, right? And to map them with STS markers or other things. And the good thing about this is that you just had to do shotgun sequencing. All you had to do was shotgun sequencing. Just build a factory and do that. One factory floor. The other thing is that I only needed 3 libraries, right? Instead of 25,000, 35,000 different shotgun libraries. Because think about it, you have to manually make a library for each back. We just had to make 3 libraries. Ham Smith, okay, made all the libraries, okay, spent tons of time doing it, could make them to the highest quality. Okay, I mean, he basically did them at, he basically would like triple size select, right? Triple purify, okay? So we had good size selection. I mean, he never looked stressed, okay? Okay, but the trade-off, of course, is that nobody thought that the assembly could be done. But there were, in 98, you know, when I went, I actually did kind of do an analysis. And before 98, it actually would have been hard to have done this thing. But some things, the wind had just changed in 98. What happened is that there was the advent of the capillary gel sequencers. Before that, the DNA kind of flowed down on a slab and it kind of went like this, you know, and it was very hard to tell. Sometimes you would cross the lane, so it was very hard to tell if a pair was a pair. because you were making lots of lane mistakes. So pairing fidelity was very weak. But with a capillary, where they flow down the capillary, you knew exactly where each sample came from. So that meant that the lane tracking problem disappeared. It also was a big gain in automation. Another really important thing that you young guys may not know is that we didn't have good 64-bit machines until 1998. The DEC Alpha, when we tested it, the DEC Alpha was the only 64-bit machine that would stand up, okay? All right, IBM came in a year or two later, but so we used the DEC Alpha. There was a lot of robotics and QC was coming in, and again, I already mentioned the issue with the library, but those were all very important things that had changed at that point, because without a 64-bit architecture, we could not obviously have done it. So let me tell you just basically how we did it, because remember what the manuscript, what the simulation said is that if I could detect all the unique stuff in the genome, right? I could get it, right? So how do you do that? Well, the idea is that what you do is we do the chordal graph collapsing, right? The thing I did in DIMAX, okay? I looked for all of the things that basically collapse into single paths, right? And then I did statistics on those to see if the arrival rate looked consistent with being single copy or multi-copy. And in that way, I could basically get every unique stretch of the genome, okay, with very, very few errors. I figure I made 10 to the minus 8 errors, and since there were only 10 to the 6th objects, okay, it was a pretty good bet that I'd made no mistakes. Then what you do is you take the mate pairs and you link them together. And you don't ask for just one link, because a link could be wrong with a 0.5% probability. But if you ask for a pair, where both of the reads end up in the thing, that's an odds of like 10 to the minus 6. Against the context of the entire genome. So then you create the scaffolds. And then once you've got the scaffolds, you can even get the repetitive stuff by what I call throwing stones or backfilling. So you take the mate pairs where one read is in the unique stuff, so you know it belongs there. And then if the mate pair places the other read in the gap, then you know that that read belongs in the gap. So you take all the reads that go in the gap, and what do? You do a little subassembly. And hey, as long as it's not a microsatellite, if it's an interspersed element that's like something way over here, it assembles just fine. Okay, so we ended up, basically doing I think a very good job. So basically because we had this factory, I mean, we basically started with 10 people in a building with nothing in 1998, and by 2001, we had basically built up the factory and done all of these assemblies, and while there was a lot of argument about how good they were, the point of the matter is that everybody does it now, okay? And I think, I think really the important thing is that really this was, and I'm very happy to have been able to done this, is that it was really, it was a case where informatics clearly played a pivotal role, right? Because nobody thought that us geeks, us guys, right, could do it, right? Because it's pretty easy to come up with an 80% solution. I'm already down to 10, am I? Okay. All right. So it involved a novel protocol and a solution to a daunting problem. And I'm down to 10 minutes? Okay. I'm gonna jump way ahead then. Okay. This was kind of a fun story, but you'll have to catch it in the bar. All right. I think I think I'll do this one. So then after the genome, okay, what happened? This decision was that cost would become the priority. Everybody wanted the $1,000 genome, right? So this is 2003, 2004. So everybody was working towards this, and the net effect of that is that today, or until recently, we've had nothing but short reads, okay? Which intrinsically means that we're doing counting and resequencing applications, okay? And there have been some attempts at de novo assembly, but frankly, as far as I'm concerned, they were never what I intended. In fact, I think it's just amazing that we went from being completely skeptical of it, right, to the point where we're so accepting of it that now it really is insane, okay? All right? What a lot of people are doing out there is insane, okay? It's not the genomes I ever wanted you guys to produce, okay, or ever proposed, okay? We shouldn't be doing this, okay? But it's all we got, because we're after the $1,000 human genome, right? Because we want to do medicine. But I want to do science. I want to understand comparative genomics. I want to understand evolution. I want 1,000 genomes of different creatures that are of very high quality, so we can do really great analysis, right? So most results truly are Swiss genomes today, okay? And I think that it's more important than ever. I mean, for example, I proposed, this 20 flies project in 2003, right? And we were going to sequence all these things. When you actually look at these things, here's melanogaster with an N50 of 21 million. The other fly, this is a good one, 180,000. A worse one, 20,000 base pair contigs. I don't want to do comparative genomics on that thing. Okay? I'm not going to understand the evolutionary history or large structural variation looking at that thing. I might understand something about how the genes and the proteome evolve, but that's it. I'm not going to really understand evolution or genome rearrangements. And if you look at one of these 100x NGSs, 8 kilobases. Ugh. Okay. All right. Now, need I say more? So I think that the emphasis on cost over reconstructability was a bad policy for science. I've already stated that these things, I think that, and if you think about it, DNA sequence is the foundation of what we do. So why should we not have perfect sequence? Okay, at least that, right? I mean, you guys deal with all the caveats, right, of all the crap that comes out of the technology all the time, right? You probably spend 75% of your time dealing with the caveats instead of actually doing the analysis that you want, right? So could we at least have our DNA neat? Okay, I'd like DNA to be neat. Okay, we should be able to do at least one thing. So so I got very excited about the PacBio sequencers. Okay, and I'll just explain briefly why I got sequenced, and I think I'll probably wind up. Okay, so you guys I think know about it. They watch DNTPs get incorporated in polymerase molecule. They have very high noise. Okay, 15% noise, mostly insertion noise, but very high. But here's the thing. Mike Hunkapiller talked to me last April, my old boss, and was telling me about this machine. And I got totally stoked because he used the word random a couple of times in key places. He said, first of all, that the errors are random. Yes, there are lots of them. But they are the results of basically thermodynamic noise, okay, and therefore very random. It's close to a random number generator as you get, okay? And the read sampling protocols, because they just involve sonication, are very, very close to Poisson, much, much closer than anything else. So these two key things are random, okay? So I got so excited I bought 2 of them last year, okay, and I'm doing some stuff in secret that I won't tell you about until I'm done. And why did I do that? So thanks to Lex Niederbrock, I actually started tweeting in February after the AGBT. He shamed me into it, said, old man, you got to tweet. So I tweeted, and I tried tweeting something probably a little bit too complicated. So I tweeted the following 140 character theorem, okay, which is that perfect assembly is possible if and only if the sampling is Poisson, the errors are random, and the reads are long enough to solve repeats, okay? And note that the error rate is not needed, is not involved, okay? And just very quickly, I mean, the point that matters, if the sampling is Poisson, then it means that if you do enough sampling, you can cover the entire genome to any degree of depth that you want, okay, provided you sequence enough, okay? And if the sequencing errors are random, then if you start doing consensus voting in basically multi-alignments, the accuracy converges geometrically and there's no limit. That's why you see with, for example, the PacBio, you see Q60, right? You never see better than Q40 with any other technology. Why? Because once in 10,000 base pairs, there's an error that occurs in every single read because it's reproducibly making the same mistake. Whereas if the errors are random, there's no limit to how accurate you can be. And C is a bit soft. I was trying to be controversial, right? But hey, if 80% of your interspersed repetitive elements are less than the size of your reads, and remember, these reads are 10,000 base pairs long. They're as long as my mate pairs at Celera. So now I not only have the outside, but I have the inside, okay? Then you basically can get it, okay? And so I got very excited about this instrument. If you give me another instrument, I've defaulted, I have 2 of them. If you give me another instrument that had the same characteristics and was cheaper, I'd use it, okay? But this is the only one that has these characteristics, which is why I'm going with it. But the problem then, okay, because that's a nice theoretical argument, is how can I do it with as little coverage as possible, right? And how can I do it efficiently? Because at these error rates, these computes take tens of thousands, hundreds of thousands of hours if you're not careful, okay? So it's really, the problem is an efficiency issue, both in terms of computer time and space, and it's an efficiency issue in terms of how much coverage do you need in order to get the result that you want, right? Because right now, everybody, for example, is doing 50 to 100x. But what if we could get, in principle, we should be able to get the same result with 30. Why not? It's our game, guys. We're the ones who are going to make it happen, okay? So I was going to give another whole talk, okay, on my work on PacBio, but I'm not going to have it. So just know that I started a blog. It's called the Dazzler blog. You can go get my software, okay? You can see what I'm doing with my colleague, Siegfried Schleusnisch. You know about Jason's stuff. So basically, we've, I guess just to kind of wrap it up, I think we've solved the speed problem. We've basically got something that's 25 to 45 times faster than what's currently available. So for example, the human genome was done with 404,000 CPU hours for the overlap stage. We can do it in like 14,000 CPU hours. Okay, we can get the entire human genome done in 4 days. We can do a fruit fly in 300 hours. Okay, CPU hours. So literally a couple hours elapsed time. And we're working on a number of ideas. I've also worked on storage. I can basically, you know how the bane is you have to store all these terabytes of these weird esoteric files that you get from the company. I can pull all that stuff up and losslessly compress it to give you a factor of 14. So instead of storing 2 terabytes for the human data set, you can store everything you need in 150 gig. Okay, here are some speed comparisons to the previous tool. It's more sensitive. It uses some ideas that are going to appear in a Wabi paper in September 2014, so I'm going to disclose all in terms of how I got the speed. And I actually put it in xArchive as soon as I've written the kind of an additional chapter section that I want to write. I need to wind up, don't I? Because this was such a nice point, Gerhard. Okay, anyway. This is actually from my 2005 paper. This is actually the graph that you get. I'll make one more point, all right? Because I think this is a good one for all the assembly geeks, okay? So this is in 2005, I wrote a paper. I showed how to kind of build the string graph in linear time, or how to do the transitive reduction in linear time. And this is the result of taking the actual C-Jejuni sequence, but simulating the reads, okay? And when I simulated the reads, I got this beautiful string graph, okay? Beautiful, exactly what I wanted, right? Just like the picture I showed you earlier, the cartoon I showed you, okay? But when I actually took the real data and ran it, it didn't look so good. It was really fractured, okay? So why did it have to be fractured? Well, it had to be because the conditions that I was assuming in the simulation were not true. So what were the conditions in the simulation? The conditions were that the reads are actually contiguous stretches of the underlying genome, right? That each read actually comes from that genome and is a contiguous stretch of that genome. And the other one is that the reads were not too erroneous. They were, 2%, 3% perturbations of the genome. So what must be wrong then with the real data set? Well, the real data set has chimers in it, right? It has DNA that doesn't come from the genome. It has unclipped primer sequence, right? All things that violate the first condition, that you're a contiguous stretch of the genome. And it had excessively erroneous reads. It had some real clunkers, where Fred said they were good, but they were not good, okay? So the conclusion is that, and there's been a lot of work in the community on kind of building what I call scrubbers, where you actually look at the data and you try to fix it, right? You try to anticipate and find these things, right? And I think it's underappreciated. I've seen a lot of work, I've seen bits and pieces of it, but I've never seen anybody go after it. I think that that's the real limit to good assembly. And so what I do is I actually find all local alignments. So the deliner that I'm talking about, it doesn't just find overlaps, it actually finds every significant local alignment. And so you see things like this, right? Where that's clearly a chimer. Or you see things like this, where you clearly see a repeat. So with these things, we're working on a thing. The other cool thing that we can do is we can take every 100 base pair stretch of that genome. So these long reads are really noisy, and if you look at their quality values, they don't tell you a darn thing. Useful for actually producing a multi-alignment, but in terms of estimating, is this 1,000 base pairs good data or bad data, it's uninformative. But if you actually look at how a read lines up with the other reads, and you look at 100 base pair segments, okay, you can see the places where nobody supports that read or not. And in fact, I color them red, okay, so that it's a heat map where the redness is proportional to the degree of difference between the reads and that 100 base pair stretch. And look at it, you can see it, right? You can see that that basically there are a few regions there where the underlying read has got to be really crappy, right? The segments that I've lit up there, okay? And in fact, if you do the complement, you can see, right, exactly how to fix it. So you get exactly, so for example, you look at this one, you know you need to trim the end of that one, right? Maybe you need to throw this one out, okay? So what I'm hopefully getting at is that you can see these things. So we can do a bacteria in 15 minutes on this laptop, actually. We can do Arabidopsis and flies and the human genome. And frankly, I'll put it this way to tell you in terms of what I'm trying to sequence. The human genome is a warm-up. Okay, it's a warm-up. And we think that there are going to be a lot of processes. So, I mean, and we're just really like excited. Like, here's E. coli. The assembly is a circle, just what you expect. And in fact, in our newer version, it's an even tighter circle. There's no kind of breaks or divisions. This is Aetheliana. I mean, we have a 13.5 megabase pair genome. So let me conclude then, which is that high error, as long as it's truly random, is only problematic with respect to compute efficiency and the coverage required. It says nothing about the quality of the result that you can produce if you're determined enough and you have enough budget, okay, which is the one thing. But the cost on these things is going to come down, okay? If Nanopore ever gets its thing to work, it's going to be cheap. And so, and I feel that at least to date in the last year, we've solved the efficiency issues. We're happy with our performances. And so our goals now are to, like many other teams, are to really produce superior assemblies and to assemble with less, okay, 20x, 30x, not 50x, okay? So I think, I'm very excited about long-read sequencers because I really do think that they're enabling the resurgence of de novo sequencing. And I think that, you know, we're really going to be able to go out there and sequence the world, okay? We'll be the Linné of our times, right? And we'll go out there and be able to sequence the world and really produce stuff that's like of such a high quality and so complete that you guys are going to find all kinds of cool things, okay? All righty, so I want to thank the few people that have been doing the DNA sequence work, and thank you for your attention. Auf Merksamkeit.
Thank you for your fantastic talk. If you have questions, please step forward to the microphones. They are in the aisles in front.
5 minutes to the game.
Germany never scores early, and not as Argentina. So, time for questions. That is the game.
It was too clear.
Funny. Bonnie.
So you're talking about, you know, ab initio folding. What do you think about using reference genomes to correct for the errors when you have tons of individuals in a reference genome?
Yeah, well, so the point of the matter is and Evan Eichler really is the one who makes this point, which is that we don't really understand large-scale structural variation in the human genome because we can't see it with the current technologies. And it's clear that there are large-scale structural variations. I mean, 1 in every 100 births, there's a huge chromosomal rearrangement, okay, that leaves the individual completely fine, okay? And also, I think in terms of the Cancer Genome Project, okay, I mean, we need accurate understandings of copy number, and we really need to know how it's scrambled, because a lot of the scrambling's happening at the macro scale, not at the micro scale. Right? I mean, yes, there's some key point mutations that set things off, but if you want to understand how the thing's been blended, okay, you really, I think it would behoove us to actually do a careful whole genome shotgun of some cancer genomes. Does that make sense, Bonnie? Did I answer your question?
That made sense.
Yeah.
I'm just not sure how much variation there is in the human genome, but I'm willing to look into it.
Yeah, and I think...
But cancer genomes, I agree.
Well, I think there's another reason why we want to be able to de novo sequence, which is that, you know, with new technologies like CRISPR, for example, it's very easy to develop model organisms now. It's getting easier all the time to take a critter that has a fantastic phenotype, like, say, completely regenerative, okay, and make that your model. But you need a genome, right, in order to do the CRISPR and the transgenics.
Yep.
So, but I mean, imagine we can take a regenerative creature and then find some guys in a very narrow clade nearby that are not regenerative, right, and do comparative genomics. So biologists should be able to pick their organism according to the phenomenon that they're interested in studying. I mean, I think we can turn this thing on its head. Instead of saying, oh, I have to do it in a fly, because that's the only thing I can do genetics in, we can turn this thing completely around. But we've got to be able to sequence at reference quality in order to enable the models.
Thank you for a very inspiring talk, Gene. So I'm by no means what you called a assembly geek, but I do assemble genomes with a gun to my head. And what I sometimes find, and my colleagues find, as non-professional assemblers, so on, that it's still an incredibly hard task when you do de novo assembly and you get to the finishing stages, and you want that clean genome that you so strongly advocate, and I totally support that. Due to repeats, I guess, mostly, and other issues, which I sort of missed in your talk. Would you care to address that? Because even if the error rate is low, good assembly, and I'm only doing prokaryotes, I have an easy life, is still incredibly
I sympathize, and in fact, it's one of my big frustrations in this too, which is that I felt like, you know, at Celera we took it to a certain level, right, where we could get assemblies of a certain quality, and we don't seem to have really made significant strides in terms of resolving repeats and finding these caveats and solving the problem. And I cannot explain to you why that's true. I think part of the problem is that there's different phenomenology. In other words, each time the technology changes, the caveats change. And so it's very hard for the developers to keep up with that. And then I think the other thing is that just we have not, this is kind of why I'm back in the game, I'm dead set before I die, okay, to solve this damn problem, okay? Let me put it that way, so that you're a happy man. My goal is to make you a happy man before I die. Okay?
I would like to postpone that date as long as possible, but I would like to be a happy man as short as possible. So, but do you think that I was hoping that you would say something like maybe the long read technology would solve this repeat issue, because that would seem like
it certainly helps. I mean, we're getting stats that are really phenomenal. I mean, we're getting stats that are better than the Celera stats for the first time. You know, Adam Philippi just gave a talk, and his N50s are basically almost equivalent to the current release, and they actually fill, and we're getting similar results. I mean, we're getting 20 megabase pair N50s. Think about that.
Well, you already made me a happier man, but please
yeah, no,
living.
So these long-read instruments will really change things. No, sorry, it is getting better, but it's still the case that there are certain combinatorial aspects to repeat resolution and identification, which I intellectually understand, and I don't understand why we can't get them into an assembler in a way that works yet. And so I'm dead set on doing that.
Thank you very much.
Yeah.